Crohn's disease, an immunohistochemical study written and directed by Ashley Penn. What is Crohn's disease? Crohn's disease is a type of inflammatory bowel disease, or IBD, that causes inflammation of the lining of the digestive tract. In the study, the background shows that case studies in the past repeatedly suggested that the fundamental alteration in Crohn's disease occurs in the regional lymphatics of the intestine. Immunohistochemical study, the question that the research is trying to answer and the aim of the overall research is the evaluation of the lymphatic inflammation in Crohn's disease and the characterization of lymphoid aggregates and granulomas in and surrounding lymphatics and blood vasculature. In the duration of active Crohn's disease, the hypothesis was the intestinal lymphatics are the base of Crohn's disease in men and women. The predicted results were that in a study of 23 control patients and 24 Crohn's disease patients, the amount of lymphoid and granulomas in the Crohn's disease patients would be higher than those without Crohn's disease. Patients and tissues consisted of 48 tissue blocks from 24 Crohn's disease patients and 23 tissue blocks from 23 control patients. Tissue sections were immunostained and their labels were lymphatic endothelial cell markers, blood vasculature markers, T cell markers, B cell markers, and macrophages. Patients were both female and male, aging from 14 years of age to 60 years of age. They show the demographics of Crohn's disease patients and demographics of control patients. The experiment was tested by utilizing different controls that were tested in the tissues within the control and experimental groups, and the ileum from normal patients going through a colectomy from colon cancer, normal patients undergoing surgery, and normal ileum from car accident trauma were depicted in the demographics of the control patients. The methods of immunohistochemistry, these primary antibodies were heated, cooled, steamed, chilled, held at room temperature, and then set for a final wash buffer. After their final wash, these slides were constrained and approximated positive for human tonsils and the colon and negative for emitting primary antibodies. These controls were run with each other and for another batch of 20 to 40 test sections. The methods for scoring of lesions consisted of 23 histologic features of disease for the mucosa, submucosa, muscularis, serosa, and the subserosa. Also, for freestanding granulomas, those were labeled and scored as 1 because scoring was labeled based on a 0 to 3 per tissue section. The results seen in Figure 1 show the mucosa for the control patients and the Crohn's disease patients, identified by the black arrows seen in subsection A, B, C, and D. The results for Figure 2 show different types of lymphocytic aggregates in the ileum of Crohn's disease patients for subsections A, B, and C of the ileum. The results for Figure 3 show the lymphatics being filled with lymphocytes as seen by the arrow in subsection B and without lymphocytes as seen in subsection A. These were both found in the ileum of a Crohn's disease patient. The results for Figure 4 show a submucosal lymphatic in subsection A and confirmed by the occluding granuloma in subsection B. This is depicted by the arrow and are confirmed by CD68 staining. The results pertaining to Figure 5 show in the ileum of a Crohn's disease patient freestanding granulomas as seen in subsection A on the left and non-freestanding granulomas as seen in subsection B on the right. Lymphangiectasia and lymphocyte paralangitis were demonstrated in all 24 patients, lymphocyte obstructed lymphatics in 7 patients, granuloma obstructed lymphatics in 9 patients, and inflammatory lymphoid follicles in all 24 patients. Freestanding granulomas occurred in 19 patients, and in 3 further patients, granulomas were in or attached to blood vascular units. The study supported the hypothesis that intestinal lymphatics are the base of Crohn's disease in men and women. Also, the study supported that the predicted results in the study of 23 control patients and 24 Crohn's disease patients, the amount of lymphoid and granulomas in Crohn's disease patients would be higher than those without Crohn's disease. Data can be interpreted by the 24 Crohn's disease patients as a demonstration of lymphocyte-obstructed lymphatics, as seen in 29% of the patients, granuloma-obstructed lymphatics in 38% of the patients, and all 24 of the patients with Crohn's disease had inflammatory lymphoid follicles. A closer look into the types of granulomas occurring within the patients identified 79% of the granulomas as freestanding, while only 13% of the patients had granulomas joined or a part of their blood vascular system. These results were significant because they defined a different subparts of Crohn's disease and how they associate with the inflammation of the organs relating to Crohn's disease. The study showed the different classifications of Crohn's disease and how the illnesses can be characterized into two unique categories that helps determine the treatment of the disease in particular person. Dr. Sura, Dr. Collenbowl, Dr. Van Krugen supported their aim of evaluating the different classifications of lymphatic inflammation relating to lymphatics and blood vasculature in Crohn's disease. This is because their evidence shows that 29% of inflammation was due to their lymphatic structure and 38% of patients' inflammation was due to the structure of their granuloma. Their findings from this experiment helped them discover an understanding of the inflammation patterns in Crohn's disease patients.